My name is Ali, a PhD student at TU Dresden. I'm working at bioinformatics groups at Biotech. Well, today I will speak about our project in pancreatic cancer diagnosis, and by end of the talk, you will know how we used deep learning on genomic data to improve pancreatic cancer diagnosis significantly, in particular in differentiating pancreatic cancer from chronic pancreatitis. Well, speaking about the currently available methods or the most intensively used method, CA99, a carbohydrate antigen, it's a biomarker that is usually used as standard biomarker in the hospital for evaluating pancreatic cancer. So this antigen is released by cancer cell and can be easily detectable from blood. With evaluating the value higher than 37, it is an indication of cancer. Lower than 37, it's kind of a green zone. Nevertheless, it can be also elevated in other diseases rather than pancreatic cancer. For example, chronic pancreatitis, benign tumor of pancreas. And this is a reason of false positive diagnosis. And a false negative coming that there are some people missing CA99. For example, 10% of Caucasian missing this. So overall, the accuracy of CA99 around 80%. Thinking about other methods, for example, imaging endoscopy biopsy, you should also consider with the higher accuracy, the high invasiveness and the higher pain. Well, so what is the alternative diagnosis method then? So people use, for example, define some biomarkers. There are lots of great studies that define biomarkers, other define mutations or variants for diagnosis. But considering the high variability of a biomarker, make it limited. And for example, gene expression, metabolic. These are kind of highly influenced by the environmental factors, by treatment responses. Variants, on the other hand, they are stable, static, but with limited applicability. There are lots of great wide association studies that define some important variants, but always the problem of a limited applicability. Few percent of patients that can be affected by this. So here in our project, we seek to exploit the synergy between these two kind of features, the variants that have lower variability and the biomarker, in particular CA99, that have a higher applicability in order to have higher performance of diagnosis. So our workflow is divided to multiple steps that involved in this project. So I will go briefly through them with these few minutes that I have. And of course, lots of details will be, but they are already included in the article that we submitted, and hopefully it will be accepted and published soon. Well, I will start with a patient that we have. So we obtain samples of nucleotide cells from peripheral blood, and we sequence RNA with a depth of 60 million fragments. And our patient cohort are characterized as the following. 2 cancer groups, one resectable pancreatic duct adenocarcinoma and the other non-resectable. And here it is non-resectable because either it is metastasis or it is for the tumor forming in the head of pancreas would make it difficult to be cured. So this is an RPC. The third group, CP, chronic pancreatitis, it is not cancer, it's inflammation. But why it is very important here to study and distinguish? Because CP sharing lots of characteristics, lots of symptoms with cancer, which make it difficult to distinguish in the classical ways. So our samples are liquid biopsy. Here, why we take liquid biopsy, not the tumor biopsy, because from tissue biopsy, I mean, because of we want to keep the minimum invasiveness, the minimum pain and risk, and the high throughput. And we wanted to see if this approach can make great contribution if we combine it with the blood biomarker later. So our samples are around 300. Fortunately, they were balanced in terms of number, around roughly 90 sample in group, and our two group, RPDAC and NRPC, they have clear difference in the survival times. So this is why we kept them always two groups. For example, RPDAC, the people survive double of an RPC. We also collected CA99 values because first we want to use them as a baseline, second we wanted to use them as features. So we apply deep sequencing and we go through long procedure of sequence assembly, alignment, variant calling till quality control. Here we had mainly we use GATK best practice in this procedure, and we apply additional steps for quality control. That we went from the 2 million raw variants to 17,000 high quality variants. Mainly the missingness affect the number of the past variants, but at the end we wanted to be sure that we have a robust system, that is when we replicate it more and more, we will give us more or less same results. Samples also, we kick out some samples either because of heterogeneity, missingness, relatedness, stratification. But they are not a lot. Most of our samples survive. In our study, we have two parts. We have the statistical analysis, which already take the genetic variant association with the disease, or with either with case or control, and the other one is the machine learning. So I will focus here on the machine learning part because of the time that I have. First of all, we take CA99 and we evaluate, and we see what is the performance if we use just CA99. So we measure the AUC. If you are not familiar with the AUC, the closer the AUC to 1, the closer to the perfect, and closer to 0.5, we are close to the random. And the AUC of CA99, it was better, a bit better than the average. The statistical analysis show us we couldn't define any single variant better than AUC. So the signals with the variance, even though we define very significant variance in terms of p-value, but they are still below CA99. So when we ask, can the combination of a variance make difference? And to combine variants, for example, 70 or 100 of variants that are significant, we use proper model for that, deep learning model, because it's able to learn complex and non-linear relationships. We apply a feature selection process on training set, and we optimize the model with a training set, then we test with independent testing sets. The result here is characterized as the following. First, we combine the cancer groups, the two cancer groups together against the CP group, the inflammation group. First, we measure CA99, and the AUC was 84%. Then we take the variants without CA99. They were around 83%, the combination of the 70 variants, and they were around 83. So they are not really better, but almost close. What make clear distinctions is the combination of variant and CA99. So we have like 96% of performance. We are very close to a perfect when we take both, and the performance became even better with RPDAC versus CP when we take only one group, the resectable group, the homogeneous group, against the inflammation. So the variants were better performance, 89, and the combination more or less as high as the previous. However, by taking the non-resectable one, the performance of the variants became lower, 76%, but the combination achieved like 92. Still good, but not as good as resectable. And the reason is the non-resectable group is more heterogeneous because some of them metastasis, some of them different kind of pancreatic cancer, either in duct or in the head of the cancer. So it is no surprise. But to sum up, our model achieved area under the curve around 96% and minimum 92 with this group. So which is already great contribution and already great improvement. And with this, I will end up my presentation. I wish you enjoyed it and thank you so much for your attention.
Okay. Thank you for the presentation. So we have some questions, and I'll just show it on stage that Jake Crawford says that it's a really interesting talk. In your input data, I noticed that the CP group was fairly different in terms of gender and age from the cancer subtypes. Did you include gender and age as covariates in your model, or do you anything else to control for them? So, yeah.
Thanks for the question. The simple answer is yes. We already noticed that there's a bias, and this bias coming from some clinical reasons. So we already added as a covariate in our feature selection and in all the whole process.
Okay. So, okay. Any more questions? Please do ask questions, and I think speakers also welcome questions, and it's makes the talks more interesting. So please don't be shy. I can. So okay then.
I can
Okay, there
already answer the
is.
next one.
Yeah, there is another question by Use from Germany. I hope I pronounced it right. Thanks for the nice talk. Which deep learning architecture did you use and why did you choose it?
Here we use deep neural network, and that is just a simple architecture with three hidden layers, and that's it.
Okay. Another question from Gaurav Ghosh, and he says it's a great talk. In your description, you had mentioned the machine learning model having a visible layer. Do you mean the input layer or the first hidden layer?
The input layer, and that is how we feed our features in the input layer. This was our visible layer in that context.
Okay, thank you. And there is another question by Jennifer. Can I use this model with CNVs, AMGG, and gene fusion?
Sorry, but I wasn't sure what do you mean with CNVs. I'm not familiar with the term. Can you clarify it, please?
Uh, uh, yeah, copy number variation. So Ali,
Oh
can you
yeah.
be a bit louder? You are not, or I hope everyone
Oh
can hear.
sorry.
Yeah.
Yeah. Sorry. Okay. Well, not really. It is not really about the gene fusion. It's more about kind of we are looking for variants that are already in the liquid biopsy, and this um, we believe that these variants are causal because we found a correlation between these variants and the level of CA19. So the cases that CA19, the carbohydrate antigen, fell in them, our variant successfully managed to handle them. So this is how our model exploit this relationship in kind of 5 or 6 variants. Other variants were associated with chronic pancreatic, so that is kind of inherited, and we already find some of them in GWAS study. So it is more about the variant level, not really the gene level. I hope this answers your question.
Oh, yeah, I hope your questions were answered.
Still
If
one, I guess
anyone,
we skip it. There is one question about single cell. It is not single cell. This is deep sequencing with liquid biopsy. So just to answer the one that we haven't covered.
okay.
I don't know
So
what was the name, but I remember the question.